Hello students, this is Dr. Faizan Mirza. In this video, I'm discussing with you the expected answers because the MS is not yet out of Feb-March 2026 paper. Variant 2, Biology AS. The first question they have simply asked you to put a tick or cross for the animal and the plant cell regarding the feature of ATS ribosome. We know they are present in both. The cell was composed of peptidoglycan, neither. Large permanent vacuole, no for animal cell, yes for plant cell. The next part shows to you phloem sieve tube and a cell A, and you can see this sieve plate is there. They are showing to you a part of the cell surface membrane of cell A. We know cell A is the companion cell. So this companion cell is having the plasmodesmas, various plasmodesma that allow sucrose to enter into the sieve tube. This particular cell membrane is having a proton pump. We know the proton pump can push the H plus ions out into the apoplast pathway, and the co-transporter protein then allow the same H plus ion together with the sucrose to enter back into the companion cell by the secondary active transport, and the H plus can then go back again by active transport. So H plus goes out by active transport. We are using ATP breakdown, the energy released from ATP breakdown, and the sucrose H plus co-transporter brings it back. So if there are more co-transporters, so a lot of sucrose can get moved in like quickly. Describe and explain how the structure of cell A is related to its function of transporting sucrose into the phloem C2. So we'll just mention all this thing. The cell is contained cell, proton pump is membrane, use plus push the H plus ions out into the apoplastic path through the cell of the active transport. This established the electrochemical gradient across the cell surface membrane, and the H plus ions. The co-transport protein uses the electrochemical gradient to transport sucrose back into the companion cell against the gradient, whereas the H plus ions are moving down the gradient. So this becomes a very important point that one of the things is moving down the gradient, the other is moving against the gradient, like sucrose being actively pumped in, like pushed in, like pulled in, sorry, not pumped in. Sucrose enters into the C tube element by crossing the plasmodesma down the gradient, while the H plus ion moves out again with the same proton pump. We can add another point of many co-transporter being there and many plasmodesma being there. So this process occurs at a fast pace because it is shown to us in the diagram. Two of them are shown to you. We'll use this as a hint. Multiple plasmodesma are shown to you, so we'll use this as a hint. Explain how the sucrose that enters into the phloem sieve tube from cell A is transported to the sink. So we know that sucrose lowers the water potential in the sieve tube, so water enters by osmosis down the water potential gradient from the xylem to the sieve tube element. The hydrostatic pressure gets raised and the sieve tube element and the mass flow occurs. Sucrose to sink. Moving into the sink, sucrose water solution moves in one direction along sieve tube. We mentioned that it goes along one direction, crossing sieve plates through the sieve pores. Upon reaching sink, the sucrose is removed at the sink during unloading, and this maintains the gradient. Question 2 deals with blood transports many substances, including gases. Explain how the properties of water allow this to function in the mammalian circulatory system. So we know that water acts as a solvent because water is polar. It's able to dissolve polar substances such as glucose, amino acid. It's able to dissolve. The property allows the water to form hydrogen bonds with positively charged substances as well as negatively charged ones, such as sodium ion, hydrogen carbonate ions, chloride ions. We'll give example wherever possible because the command word is explained. Then cohesion between water molecules allow blood to flow as a continuous column within vessel without breaking, and incompressibility helps maintain pressure in the liquid state, allow the blood to flow. High latent heat of vaporization allow blood to lose heat near the surface of the skin, cooling the body. Although the last point is not very relevant transportation, but yes, I will go with that. State two different ways in which carbon dioxide is transported in blood. So we include 10% of carbon dioxide transport is carbon monohemoglobin inside RBC bound to the amine group of hemoglobin, and carbon dioxide is transported 5% solution in plasma. We can go for the bicarbonate ion as well. So mentioning the percentage and all. So let's move. The next question, the next part. Here, the ribbon diagram is shown to you of a myoglobin. They're saying myoglobin is found in the muscle tissue. You can see that this is one protein made from one amine end and one carboxyl end. It means this is one polypeptide chain. There's an alpha helix. So this is alpha helix. And there are the beta strands as well. And there are some random coils. And there's just one prosthetic group with the iron ion. So only a single oxygen molecule can attach to it. They're asking you describe the similarities and differences between the structure of myoglobin molecules shown in figure 2.1 and the structure of hemoglobin. Recall this hemoglobin. So both hemoglobin and myoglobin have alpha helices, beta pleated strands. They both contain heme groups. Prosthetic groups, they both have carboxyl and the NH terminals on opposite ends. Myoglobin is having one polypeptide chain, hemoglobin is having four, and there is one heme group in myoglobin. There are four in hemoglobin. Myoglobin is having one oxygen molecule that can binding site, whereas hemoglobin will have four oxygen binding sites. Contracting muscles have a high requirement of oxygen, which is supplied to the muscle cell by hemoglobin. We see myoglobin has a higher affinity of oxygen than hemoglobin. So a higher affinity of oxygen for hemoglobin suggests why it is important for the muscle function that myoglobin has a higher affinity. Why not lower affinity? Because if it had lower affinity, it would lose oxygen before hemoglobin could lose. So this ensures oxygen is transferred hemoglobin to myoglobin and then to the muscle, and not the other way around. Like hemoglobin passes on the oxygen to the muscle and not myoglobin. Myoglobin acts as a store of oxygen and can provide additional oxygen only when the contracted muscle is respiring at a very high rate and needs a lot of supply of oxygen beyond which the aerobic capacity can deliver. So myoglobin release unloads oxygen only at very low partial pressures when the demand of oxygen is not being met by oxyhemoglobin alone. The next question deals with raffinose, a sugar found in many plants. They are saying this is the structure of raffinose. It is formed by three different monosaccharides, one of which is glucose. On the figure, draw a part with that can be hydrolyzed to form glucose. So we know that this part can be hydrolyzed to form glucose. So we just encircle it. It needs to be terminal. This cannot form glucose because it's not the six carbon sugar. It is a six carbon sugar, but there are like four carbons in the ring and two carbons outside the ring. So we need to have a ring which is having five carbons in the ring and the sixth outside the ring, which is glucose. So which type of glucose form? We know alpha glucose form the glycosidic bonds here. Number of glycosidic bonds in one molecule, reducing sugar, not reducing sugar. So glucose is having no glycosidic bond. We know that it's a reducing sugar. Maltose is having one bond because it's made from two glucose units. It's a reducing sugar. Raffinose, it's having two glycosidic bonds and it's a non-reducing sugar. Some bacteria in human digestive system has an enzyme known as alpha-gal, which breaks down raffinose. So this is a breakdown of alpha-gal by alpha-gal shown to you. So again, the Vmax can be read from here. Half Vmax can be tabulated, like just calculated by dividing it by two, and you can just drop it down on x-axis to read the value of Km. Again, you know how to read the axis. You can just do a simple conversion. How many squares represent how many units, and come up with the value here. You need to make sure that you don't miss onto the unit when mentioning the this particular km value. So this will be the km value here, and this by mistake I've mentioned arbitrary unit. These are not arbitrary unit. This is mole dm cube. Okay. One molecule is formed by breakdown of raffinose, as mentioned to you, and which is this? This is a fatty acid. We know it's a fatty acid because it's a carboxylic end, and there's a term three carbons there. The molecule shown in figure 3.3 can be used to make a triglyceride. Describe how this molecule becomes part of a triglyceride molecule. The fatty acid can react with glycerol. Glycerol, we know, is a water molecule removed by condensation reaction. OH is lost from the carboxylic acid and fatty acid, and H is lost from the OH of glycerol. These both are joined together by ester link. So glycerol is having three of these. So a total of three fatty acids can attach, including this one. So a total of three water molecules will be removed. RSV is a virus can infect human gas exchange system. Explain why this disease caused by RSV is an example of infectious disease. This disease is called by a pathogen, and a pathogen gets transmitted among different individuals. So this must be having its own transmission cycle. That's why it's this is an infectious disease. This diagram shows you the structure of RSV. So if you look closely, protein subunits are shown here, and there is a strand of RNA. So this is the typical thing that a virus is having. Then there is an outer envelope. If there's an outer envelope, we know that envelope is made up of phospholipids. We have studied that in HIV as well. So they're asking about Q is. So Q is the lipid envelope. The structure is figure 4.1 composed of protein subunits is capsid. This is the region in the bronchus shown as X. Y is the ciliated epithelial lining. So X must be the smooth muscles. State the function of label structure Y. These are the cilia, so they move the mucus over the surface of lining of the bronchus. Then they have mentioned two forms in which this can be treated in an immunity. So injection of monoclonal antibody provide artificial passive immunity, whereas vaccination provides artificial active immunity. Active immunity is where you know that the memory cells get produced. A passive is when you receive antibodies. Outline how monoclonal antibody to protect person from RSV is produced using hybridoma method. So you inject the RSV antigen to the mice and isolate the B lymphocytes and plasma cells. Fuse these cells with myeloma cell, causing fusion using fuses in non-electrical current. A hybridoma cells form, select the desired anti-SRV antibody producing cells, allow the hybridoma cells to grow into tissue culture in lab. These clone of cells produce monoclonal antibody that can be used to protect people against RSV. State one advantage of using vaccine instead of monoclonal antibody. So vaccine is having antigen, so it provides long-term protection. Memory cells, the B and T are produced. These are long-lived cells, so they provide lasting protection. Monoclonal antibody is not an antigen, so it's just providing. There's no immune response being evoked, so it's a short-term protection only. Phagocytes can remove cells from the body that have been infected by a virus such as this. Outline the event that lead to the formation of phagocytic vacuole. So upon recognition of an infected cell, the cell surface membrane wraps around the infected cell, forming pseudopodia. Pseudopodia are those structures that form like this is the pseudopodia. So it literally this is the thing it need to phagocytize. The cell produces a mouthpart and it engulfs it and pulls it in. So it produces pseudopodia. The cell around it inflows, it invaginate, the vesicle pinch inward, the cytoskeleton moves in the phagocytos vacuole enters the cytoplasm of the phagocyte. Which cell will be used to destroy the lysosome because it's having the hydrolytic enzymes? Next question, deal with DNA replication. Describe the role of DNA enzyme, DNA polymerase in DNA replication. So DNA polymerase allow activated nucleotide to bind to the exposed base of the DNA strand, be copied by the complementary base pairing. It ensures adenine paired with thymine, making 2 hydrogen bonds, guanine paired with cytosine using 3 hydrogen bonds. This enzyme allows new strands to elongate in 5' to 3' direction both. One nucleotide is joined by the complementary base pairing. DNA polymers allow phosphodiester bonds to form along the sugar phosphate backbone. So we have to follow DNA polymerase only. We will not discuss DNA ligase here. They are saying the two strands of DNA molecule are described anti-parallel. Explain the consequences of having anti-parallel arrangement. The consequences is that one strand continues as leading, the other goes lagging. So since DNA polymerase can add nucleotide in 5' direction of the new strand, the anti-parallel arrangement of DNA strand and DNA molecule being copied results in new strand being synthesized as a continuous strand called the leading strand, where complementary to the 3' to 5' end, whereas the other strand, which is the lagging strand, is complementary to 5' to 3' end, is lagging strand is having Okazaki fragment, which are then later joined by DNA ligase. The telomere shorten on each end of the lagging strand. Then they are saying after separation of complementary strand during DNA replication, the separated strand of DNA can become twisted coiled together to form tangles. Topa, top 2A, not TOPA. This is top 2A is able to remove these tangles by 2 steps. Step 1, top 2A form a complex with the tangled DNA, catalyzes cutting of both strands. Then top 2A catalyzes repairs and the cuts being made. Ectopside is a chemical that can bind to this DNA complex at step 1, which means here. This makes the complex very stable and prevents step 2 from occurring, which means the cut will be there and the cut will not be repaired. So just have I explained why this can be used in treatment of diseases such as tumors. So this ectoposide as a drug in treatment tumors prevent DNA repair from after cutting. So DNA damage accumulates in tumor cell. This stops cell division in the tumor cells in humans. Question 6, it deals with the fill in the blank. So it simply asks for the transport protein that include that are allowing fast diffusion to occur. So those are channel proteins, the fluidity is maintained by cholesterol, and the fatty acid tails, they are like merged together, held together by hydrophobic interactions. I hope this was an easy paper, by the way. So I hope this video was helpful for you. That's it from my side.